we're waiting for the room to fill. This series, if you've been before, you know that we usually have standing room only, so I understand there are conflicting things tonight and the freeway is completely stopped, so we waited a few more minutes, but I think we're going to go ahead and start in the interest of keeping you on the schedule. So on behalf of the College of Natural and Agricultural Sciences, or CNAS, here at UCR, I'd like to welcome you to the 2015 Science Lecture Series. And the title this year is The Science of Food and Health. And I'm Jody Holt. I'm the Divisional Dean for Agriculture and Natural Resources, speaking on behalf of Dean Mary Lynn Yates, who is at a function at the Chancellor's House tonight, so she could not come. So this is the 8th Science Lecture Series. It's been a really popular event over the last years since 2009. The first year it was so popular we had 2 series, and then we've done it every year since then. And this year we have several sponsors. So the college is sponsoring the event, plus the University of California, President Napolitano's Global Food Initiative. And also we have a new institute in development at UCR called CAFE, or the California Agriculture and Food Enterprise. And that institute is in development, and it is also co-sponsoring this event. And what we hope is that this institute, which has a founding board and a list of interested faculty that's well into the hundreds, is going to be sort of an umbrella organization for our research and outreach activities related to food and agriculture on campus. And so our hope is that the eventual director of the institute will hold an endowed chair, and the chair has been made possible by a generous donation from the Johnson family. And I don't think they're here. Jane Johnson is actually a former president of the UC Regents. I just got the finger saying she's right outside. She probably got stuck in the traffic. So William R. Johnson and Jane S. Johnson, or Sue Johnson, I'm sorry, endowed the chair in honor of your mother, Jane, and Sue is standing right here. She's a former chair of the UC Regents, and what perfect timing. So our hope is that when the institute is developed and all the approvals are done, the director will hold the Johnson Chair in Food and Agriculture. So we have a lot of high aspirations for this institute. So the series this year will consist of 4 lectures. This is the first. They're on alternate Tuesdays. So please tell your friends so we fill the room next time. And
Thursdays.
Before I start, also I want to acknowledge the science ambassadors from CNAS, which are serving as ushers. They're in the blue polo shirts around the room. These are some of our best students in the college who help us with many outreach functions such as this. They help in homecoming, they help in all kinds of events. So we're really happy to involve them in our activities. Okay. So for this first lecture tonight, I'd like to introduce Dr. Norm Elstrand, a professor of genetics in the Botany and Plant Sciences department here at our college. And Norm is chair of the committee that is developing CAFE. So he's chair of the founding committee, and he will introduce our speaker for tonight.
Thank you, Jody. I'm going to take this opportunity to thank the college for continuing to run the Science Public Lecture Series. I was the first speaker a few years ago, and this has really been a great opportunity for me to sit in the audience as a member of the public and to get to know the exciting things that are going on at UCR, as well as to engage with the public. Moving on to our speaker, it may seem hard to imagine that tobacco would be a plant that would help confer health to people, but it wasn't hard for Larry Grill to imagine that. Larry's an alum of UCR. He has worn many hats over the years, but he is currently the Dean of Research at one of the many Claremont colleges, Keck Graduate Institute, and he's also a professor at one of the other Claremont colleges, Pittsburgh College. His work in between leaving UCR and coming to the Claremont colleges included the discovery that tobacco can be a great platform for making therapeutics and pharmaceutical type products. You grow tobacco as the substrate, and then you use genetically engineered tobacco mosaic virus, which decorates its coat protein with the product that you need. This proved successful for the company Large Scale Biology, which is one of the companies that he founded, and it's in the process of becoming successful for the project that he's leading in Botswana right now. So I'd like to segue directly into welcoming Larry back to UCR, and I'm looking forward to hearing his talk.
I know I'm supposed to have lots of wires hooked to me. Hopefully they'll all be hooking up. Can you hear me back there? Okay. So let me kind of introduce myself. That I am very old. I was an alum here from 1979, so I give you a little bit of an indication of my age. But I actually like what I've done. What I learned here at Riverside was working with plants, and I love working with plants. In fact, in my earlier days, I tried working with animals, and I had to kill them, and it's easier than kill plants than animals. So it made me much happier. So I went back that route. But, so let me just kind of give you an introduction to where I'm going and what I'm doing with the food area. There's a Chinese proverb that you've all heard, which is if you want to feed someone for a day, you give them the fish. If you want to feed them for a lifetime, you have to teach them how to fish. And so basically I want to use that route to actually teach the people in the developing countries how to make vaccines to save their food supply. And currently, an example of it, lumpy skin disease, that cause tremendous cattle deaths in Western Africa, Eastern Africa, and Southern Africa. And the problem with that is that death of those cattle deprives them of food. And it's a huge amount. I think in East Coast fever, we're talking about 2 million cattle die a year. Basically needs a lot of water, a lot of feed to feed these things. So what you want to do is find a way to keep them from getting the diseases that kill them, but do it cheaply. And so that's the route we're going. So my variation is I want to provide them not with just vaccines, but with a way to make vaccines and make them cheaper, faster, and it's also going to spin off here into the U.S. Because we have some good stuff coming down the line that make vaccines a lot less expensive than they are. So I want to kind of give you a little bit of that technology and let you know a little bit about what's going on. So the mission is to teach them how to make vaccines for themselves. So I'm not going to make it here and ship it over every year. I want them to learn how to do it. Then the next disease that comes up, they can make a vaccine against that. Okay, so we're working with the Botswana government. There is a vaccine institute there that makes one vaccine against foot-and-mouth disease, and we're going to teach them how to make inexpensive vaccines for other diseases that there currently are not vaccines for. So Botswana, for those of you who don't know it, is right there, just above South Africa. It's a very unpopulated country. I believe there's about 2 million people in the country. And there's about 4 million cattle. That gives you an idea of the importance of cattle in the industry as far as each person. So if you are someone who lives in Botswana, you know someone who has cattle or you have cattle, and it's part of your livelihood. So when you have to put your cattle down because of a disease, lumpy skin disease or some other disease like that, it's devastating to the family. And Gates grants now are saying that they want us. They're giving grants to go help the smallholder farmers, so that they can actually do better in feeding countries there, because we know that we don't have a food supply problem in the world. We have a distribution problem, but you can't mail it over there and get it there very easily. So really, the truth is, we have to teach them how to do it. So I have a lot of people I'm working with there, and I go there every year. I've done it every year since 2008. And I told when I like being there, I just hate getting there. It takes me roughly 36 hours of plane flight to get there, and it's not fun. I don't mind being there. I love being there, but I just don't like getting there. So these are some of the people we're working with while we're there. Again, the government, the Vaccine Institute, and the ones I've already mentioned before. Okay. I take students every year, and we let them pet the kitties when they're there. So every year we go there and take different students. That's a story for those of you who don't. I think I'm dragging a cord here. My back online? Okay. So anyway, but then we also take them out on a weekend, and so that's not a cheetah. We don't pet this one. It's a leopard, and it's not good to pet. We also have other kitties we don't pet. But we do run into these things. So it's kind of a fun visit when we're there around that country. It's a nice country. A lot of students have been involved in this project, and we have a lot of publications and a lot of work that's been done to go forward. This is the current group I have with me right now, and that mixes both those we have in Botswana as well as at my labs. I have 2 different labs at the Claremont colleges. And they all love doing it because it's really translational research. They're actually doing lab work that's going to be used out in the real field. And so that's what makes it more fun for them. Okay, so our current targets. So the diseases we're going against are actually important diseases in developing countries. So rabies is an important disease because basically rabies kills people because animals get it and animals bite people. And so it actually kills about 55,000 people in developing countries every year. That's pretty substantial, and it's avoidable. It's 100% avoidable, but we don't have the vaccines out there for them, and we can't get it to them because of the cold chain and other problems. So we're trying to overcome that. Anthrax is another one of those diseases that cattle get. A lot of cattle die of anthrax. And of course, some of you have heard what anthrax is about here in the United States. Who's heard of the anthrax here in the United States? Okay, what's it thought of right now? Bioterrorism, absolutely. So we think, now that we're getting some good data on this, we think maybe DOD might give us some money, so we're hoping for that. But anyway, it's a lethal disease. When you get it, the toxin actually gets into your cells and kills you. We found a way to make a vaccine to get you to have antibodies against that toxin. So if you get anthrax, you're fine. You'll survive it. This is one that's only in animals. That's for cattle, and it's a pox virus for cattle. And that's, if you get it, you have to put your cattle down. And no one likes to do that to the cattle because they don't have insurance there like we have. Okay, the government doesn't give them money. They lose their cattle, they lose their income, they lose a lot of stuff going on there. Rift Valley fever, another thing, causes a lot of debility in cattle, other animals too. And then finally, East Coast fever, and this one kills at least 1.2 million cattle every year, over and over again. Not a good disease, but these are all simple fixes to all of these diseases using the technology I'm going to show you, which is really kind of cute and neat. And again, it's using what I learned here at UC Riverside, using plants the whole route and not making things difficult. So right now, the traditional vaccine production method is expensive. And you know that when they said they were going to get this H1N1 vaccine out to us, it took how long? Almost a year before they finally had it coming out. And then they missed it this year. So the one we have right now is actually only good for about 25% of them. But they can't go back and change it because it takes too long to grow all the eggs, do all the inoculation and things like that. So we've got to find a different way to do it. We just got to change it. And the reality is, and I just want to point out an example. A vaccine that the Western world cares about is something called Gardasil or Cervarix, and this is for the human papillomavirus, which causes cervical cancer. And all the teenagers should be getting this so that we can kind of stop this disease. It's basically available here because we have insurance. So if you didn't have insurance, the cost per dose is roughly $130. If you get 3 doses, that's you're running $500. Which means in Africa, how many people will get it? Remember, they don't have insurance. Okay, no one. Okay, this is not available people in Africa. So we're looking at things like that as where we need to go. So here's some realities for vaccines. Basically, if you're going to make a vaccine that's going to work in Africa, you got to make it cheap because you got to get this herd immunity to stop the disease. And you have to look for the cold chain resistance, so you don't have to deal with the cold chain, keeping things frozen all the time. You need to be able to respond a lot and fast for the diseases when they first start happening. So Ebola, say. Obviously, we're a little bit behind the curve on that one, but if we could respond quickly to an outbreak, and this way we can, and I'll show you how they can do it. But the current technology we have doesn't do that. So we need a better method, okay? So Again, this just kind of reiterates what's the problem with some of our current vaccine limitations. It takes a highly skilled force. It takes companies like Boehringer Ingelheim, Genentech. They got these big fermentation tanks. They got other ones doing the multiple eggs and have farms of millions and millions of chickens to keep these eggs going. It takes roughly 2 eggs to make a dose of vaccine. So it's a lengthy product development cycle. So we need to do something different. So this is the fun part. Plants can do this. Okay, it's really neat. And we can keep the cost. I mean, everyone knows growing plants is cheap, right? I mean, I grow them in my yard, and I think some of you do too. And we know farmers give us tomatoes for pretty cheap and things like that. People buy, unfortunately, tobacco for other purposes, and that's pretty cheap too. But the good part about plants is you don't need to worry about animal, human pathogens, because plants don't have them. So, and I'm going to show you how we can actually do this in large scale because it's been done. So I'm not making up, I'm not suggesting that this is the way to go, but it's also the way we're going to have to go. Okay? So how do we do this? And this is the fun part. This is really exciting. Trust me. Okay? So here's my first thing. Plant viruses. Now, I know that anyone who's worked with plants, they hate plant viruses. But guess what? You can eat them, and you do. If you go to the farmer's market, I walk through the farmer's market, and I look, oh, geez, there's a lettuce mosaic, so I see all these plant viruses. But you know what? Plant viruses don't cause disease in humans. You can eat them all you want. And so, again, they don't cause disease. So for plants, they're bad. For humans, they're going to be awesome, and I'm going to show you how come they're awesome. Okay, so that's why I have the little dance going on there. Okay, so here's what happens. So if you get a plant virus and you inject it into your bloodstream, your white blood cells sees a pathogen. It sees a virus. Okay, what does it do when it sees a virus? It makes antibodies against it. It's going to fight that virus. Well, it's a waste of its time because that virus isn't going to hurt you anyway. Okay, so it's wasted its time. So how do you take this? Someone wants me to do an additional log on. Okay, hopefully we'll get along without doing an additional log on. Anyway, so what you want to do is take this plant virus and do something with it so we can actually make it do something good for us instead of something bad for us, okay? Oh, it's not going to let me go. No, it's click off, yeah. Somehow I got to get back onto the IT, where are you? Okay, I'm back on, I think. Yeah, okay. So basically what we can do, and think about this. So if you kind of look at this, you say, okay, you're putting in a virus. It's totally not going to hurt you. You're making great antibodies against it. What can we do to actually use this to do something to us? And so here's what we're going to do. We're going to decorate it. When I say decorate it, see, this thing's got these certain surface proteins which, you're going to make antibodies against it. What we want to do is we want to decorate it with some of the surface proteins from a pathogen. So I'm going to have to kind of show you how we do it. So I'm going to illustrate this using some, and I'm old again. I told you I'm old. So I'm going to kind of give you a different reference point. So when I was growing up, the good cowboys always wore a white hat. Always. Okay, no matter what, they wore the white hat. And so you can know that. Now, the bad cowboy, okay? Now, so this is going to represent our bad virus, the one that's going to be kind of nasty. So they always wore black hats. I don't know, anyone that's old as me and know this too? I mean, yeah, okay. The newer generation, black is cool, so even the good guys wear black. But anyway, back then, it was only the bad guys that wore the black hat. So anyway, so this is the major difference. And so so this is going to be the outside surface of our virus. So basically, the good virus, the plant virus, is going to wear the white hat. But what we want to do is make it wear the black hat. So we're going to decorate it. So here we go. Here's our good guy. And what we're going to do is we're going to use the lab technology we know to genetically engineer it. And you got to watch closely, okay? Because what we're going to do is we're going to change that hat to black, okay? Using our lab technology, okay? We're going to give him the black hat. Then it's going to go into the system and do the right thing. So I'm going to show it now in a little more graphic view on what a plant virus. So this is what I call a good plant virus, because it's bad for tobacco plants, okay? But it's good for you because it doesn't cause any disease in you, and that's what it looks like. So we're going to call that the good plant virus. Again, good because it's for us, and I'm human. It's not going to cause a disease with me, so I'm totally cool with this. Okay, here's a bad one. So this one's called the rotavirus, and it kills about 600,000 infants annually in developing countries. We don't like this one, but we'd like to make people resistant to it, so get them a vaccine against it, a cheap, inexpensive vaccine. So what we're going to do is this protein on the surface here, okay, is something that we can actually use to decorate our virus, okay, if you can envisage that. So basically, turns out that we can get the genetic material, not for the full virus. We just want a little tiny piece of the genetic material out of this nucleus, if you will, of the virus. It's not quite called the nucleus, but we're going to get that genetic information, and we're going to take that out. And it's a tiny piece, so even students can work in it without using these complicated protective areas, because we don't even get the whole virus. We avoid that. So we get that gene, and we're going to put it in to this tobacco mosaic virus. That's using the laboratory techniques, and any student who's kind of done the basic laboratory techniques kind of knows how to do this. It's kind of easy to do. So you do that and put it in there. And so here's again, the good virus. Now you got to watch, kind of like that black hat. Keep your eyes on it. Okay. Ta-da! It's been genetically manipulated to be decorated with that protein that was on the surface of the rotavirus. Now this doesn't look like the rotavirus. It's the best we can do. It's still the cowboy with the white hat that's now wearing a black hat. But it's enough that your white blood cells are going to see that surface protein and make antibodies against that. And guess what? When the real one comes in, those antibodies are going to attack that first one I showed you. Okay? So it's going to be a protective vaccine for you just by using this decorated, non-infectious virus that can't infect you anyway. Even though we decorated it really pretty, it's still not going to be good. So now you need to, so we can do this in the lab. And I have students working in the lab, and they do it, and they're doing it with all these different diseases, trying different vaccines, and they're doing tests of it in guinea pigs, so I'm back into animals now again. Okay, so but what you can do, and this is some colleagues of mine who have a way to rapidly get the virus into the plant. And the beautiful part about that is we don't genetically engineer the plant. Okay, we just do the virus. We take the plant, we dunk it down into a special little solution that contains a bacteria that has our gene. But anyway, I'm not going to go into how it's done. But what we do is just pull a slight vacuum. The leaves take up that virus. Then you pull it back out. You let it dry off. You put it underneath the hood. 7 days later, you extract it. It has massive amounts of that decorated virus on it. Okay? This is simple. This isn't big tanks. This is simple. Anyone can grow it anywhere. Right here, we're using greenhouse space, and it turns out that's kind of the ideal way to do it. So this is kind of what's going on. So now I'm giving you a picture of it. So there's a gene that exists in jellyfish that glows bright green, and it's called green fluorescence protein. It turns out, and it's fluorescent because you use a black light above your tank, your fish tank, with this jellyfish in there, and it glows this beautiful green. Well, it turns out they've extracted the gene that codes for that protein. So what we did is we put that gene into that virus, and we turned this plant upside down into the solution. We pulled a solution. We waited for a few days. Then we turned off the lights, and we turned on a black light. And this is, I mean, in the 60s, this was cool, I'll tell you. But we couldn't do it in the 60s. But now, but think about this. You could go into, now I'm going off onto a divergent, but you could go into a nice, beautiful hotel, and they have all these plants lined up along the way that have black lights on them that are glowing this green. Wouldn't that be cool? Or for those of you who have Christmas trees, you could actually put it in the Christmas tree and just use a black light and have this bright glowing Christmas tree. Okay, so this is the power of this virus to do this kind of stuff. So the thing is, once you make this, you do have to extract it, and you do have to use stainless steel and all the other way, but that's kind of what you have to do to get it pure, because you don't want to inject things that aren't somewhat pure. But the good part is viruses and any plant virologist, and I learned my technology here at UC Riverside. You just use a centrifuge and do it 2 different speeds, and you can purify it out really cheaply. So we're talking about really getting it done easily. So we did a cost estimate of what it would cost. So give you an example, the Genentech facility up in Vacaville that they built cost about $1 billion. It takes about 2,000 people to run it. Their drugs are running in the range of, well, let's see, I guess some of the drugs they're making, Herceptin for breast cancer, Rituxan for B-cell lymphoma, they're running in the range of about $70,000 to $100,000 a treatment, but they can afford it, you know. So, but we can't afford that in Africa. So using this technology, smaller group of people, we estimate the cost per dose of those vaccines for the animal and humans to be around 15 cents a dose. Substantially different. Big Pharma doesn't care about that. They don't want that. They will pay it. Our insurance companies will pay it, so they don't care about the cheap cost. But for Africa and developing countries, we need to do something, and this is what we need to do. Okay, so other companies have found ways to scale this up. In fact, I'll talk about another couple, but this is using the latest technology. So they're using LEDs. So we got these blue and these red LEDs, so it doesn't create heat, but you can have this thing going 2 or 3 stories tall of these plants that you just dunk one day, bring them out, extract them. You can have your manufacturing facility going 24 hours a day and make tons of this vaccine for dirt cheap, okay? Plants, all plants. It's pretty neat. Okay, so I don't know how many of you heard about this disease called Ebola disease, okay? It was bad, okay? And, but it turns out there's a little company down in San Diego, and they found a way to have tobacco-related plants, the same ones I use, Nicotiana and benthamina, using this exact technology to make that plant make human antibodies that can be used to treat people who already have the disease. So antibodies will go bind to that virus and take it out. So they got approved by they did this with monkeys, so they took 18 monkeys, they inoculated all of them with Ebola. Of the ones they just let them have it and didn't treat them, all 9 died. The other 9, they waited about 4 or 5 days until the disease started taking place. Then they came in with these antibodies made in the tobacco plants, and they inoculated it, and they had 100% survival of the monkey. Macaque monkeys. So really cool. So there was some humans that got the disease. They actually had a little bit of this. So 2 humans got inoculated in Liberia and then came back to the U.S. They survived. But now the FDA said, okay, let's do this for big scale. So these companies have been making enough of this to start treating Ebola patients. The problem right now is Ebola is going down. We need more people, more patients, but you know. Well, you have to go with the flow, okay? Okay, so I'm going to go to another story because this one's actually kind of in the same realm of there's more things than just vaccines. I like the vaccines because that's my African side of life. I like anthrax because that's where I might get funding to keep this going, okay? So at the Department of Defense, they like that kind of stuff. So. But this is another one that we did work on that's really very, very interesting and works quite well. So there's a disease called non-Hodgkin's lymphoma. It's been on the rise. I think it's 60,000 people get it every year, but kind of not so good. So think back to that decorated virus. Now it turns out B-cell lymphoma patients, their cancer cells have a very specific protein on the surface that you can go and identify. You know exactly what that protein is. So we'll call that the black hat. So we're going to actually basically decorate the virus to carry this black hat from the cancer in the same kind of way. And then we're actually going to teach your own body to use its own immune system to fight the cancer that's been ignoring, because we still don't know why your immune system ignores cancers. We still don't have a clue. It just for some reason says, oh, okay, I'm letting you go. Well, we want to reteach it how to do this. So that's the thought process here. So what you do is you get a biopsy from the patient. You take and you find out what are these proteins on the surface of the B cells. You identify them. So that's just technology that every student knows how to do. You can't do what's called cloning, but you're just cloning these DNA sequences. Then you actually make your viral vectors kind of using this standard technology we have here. Then you grow them up. You have to get enough for a patient. Turns out it takes a table of like maybe 100 of the small plants per patient. You extract it, purify it, and then you put in a vial. This, from the biopsy to the vial to go back into the patient, takes 4 weeks, okay? So that's the cool part about it. So you don't have to wait forever for a drug to be developed. And this is for you, okay? Patient-specific, your cancer-specific, okay? It's not just for the whole world. So it's for you. So then you inoculate the patients once every month. So it's like a vaccine, if you will. The patient gets antibodies against this black hat, okay, or the proteins on the surface of its cancer cells. And so those proteins that you've made, these antibodies, go bind to this cancer cells on those they've been trained to do, and it kills the cancer. Okay, so we did clinical trials, and FDA-approved clinical trials. Sometimes we didn't use an adjuvant. There's things we could have done, but we only had 80% cure rate. I was just really searching for 100% cure rate. When I say cure, I mean it's gone. Okay, your own immune system, no chemotherapy. You didn't have to lose your hair, all that kind of stuff. So it's kind of site directed. So it's kind of a neat thing. We had television coverage of some of these data results, and what was the hardest part is on their television coverage, they had this thing, tobacco being a possible cancer cure. You wouldn't believe how many people called up and said, "Can I get some of those cigarettes that cure cancer?" Yeah, you see those pictures? Yeah, I want those cigarettes, yes. Okay. Anyway, so we worked with, this idea was developed by Ron Levy. He didn't know plants could do this, though. He was using a very complicated system, cost him about, he could do only like 20 patients per year, cost him a lot of money. And he said, "You plants can do this?" I said, "Sure." So we worked with him. Turns out you never get to know who your patients are because the FDA keeps that all a big secret. They're given a number. But this woman, Ali Jen Corelli, thought this was so cool, she went public about it. And patients can do that. That's up to their choice. I can't find out who she is, but she can find out more about my drug. So they go on that. So anyway, we kind of did the same kind of technology that I showed you a little earlier. She got the inoculation every month for 6 months. Anyway, it's been over 10 years now for this one, and she's cancer-free, as were 80% of the patients that we had. So kind of cool situation. So anyway, this still goes back to we're working on using plants to do all these neat things. But the one goal that I'm doing right now is I'm working with Africa. So it's my goal is to first do one battle at a time, and the battles. And actually, I shouldn't say one battle at a time. The battle is making vaccines cheaply, quickly. But I'm going after several different diseases because I have lots of people who are very excited about this and love doing this. So that's kind of where we're going with it. So this is on the campus of University of Botswana. It's in the center of the campus, and it's called Motole Moto Homo, and they speak Setswana there. It means one person, one beast. And the reason why this is really neat is because when University of Botswana got started, it was a country that didn't have a university, and they always had to send their students to neighboring countries to get their education, their elementary school education. They had to go elsewhere. So they told everyone, remember cattle's big in there, remember 2 million people, 4 million cattle. They said, "We want every farmer to donate one of your cattle, and we'll use that to build a campus." And so that's why it's Motole Moto Homo. Everyone donated a beast, and so they built the campus on that. So that's the start of the University of Botswana. Okay. I think I probably bored you enough. But I also didn't want to go over my time, and I probably went under my time, didn't I? But it leaves plenty of time for any questions of that.
Okay, that was pretty awesome talk. I'm going to handle the Q&A. Is this working? So Larry's available for questions. And if we can just hang on. We've got the science ambassador is here with. Okay.
They'll bring it right to you.
We need
I don't want to.
because we're being taped for the University of California's Global Food Initiative.
You're going to be famous.
Oh, no, thanks. You're going to be famous. So we want all the questions on tape.
This isn't really exactly on topic, but on the non-Hodgkin's cancer, is each individual have a different outer structure?
Yeah, so the question is, for these non-Hodgkin's lymphoma, does each patient have a different cancer? And the answer is yes. So there is a drug out there called Rituxan made by Genentech, and it goes against one of the other proteins that all your B cells have. And so it kills all your B cells. So if you get that drug, you have to stay away from people who are sick because you've just killed your immune system for a certain period of time. This one, we actually get the sequence. It's only for that cancer. And it turns out each person in your body, you have hundreds of thousands of different B cells. And the one that goes wacky, okay, or the one that becomes cancerous, that's wacky is a scientific term. So the one that goes that becomes cancerous is different in every B cell patient, lymphoma patient. So the odds of them having the identical surface protein is virtually negligible. Okay, so you're really going patient-specific, cancer-specific in that case. So good question though, Jody.
Okay, so I'm really way off topic. But, do you think there's a potential in the future for this to be used sort of for the other situation where somebody gets an organ transplant and has to stay away from all illnesses because of the possibility of rejecting an organ? Could you make a transplanted organ friendly to your immune system? Or is that way too far off?
Whoa. Okay, so the question here is, can you actually use the technology we have to teach your body to not reject a transplanted liver or something from something like that? I don't know. I know that there are ways to bind to surface proteins, but they're going to be degraded with time. So I think it's going to be a challenge, but it's a great idea. I like thinking outside the box, and I'll have to keep thinking about that, because if we can, that'd be cool. And then you could take anyone's organs. You wouldn't have to be matched. You could actually find a way to cover them. But yeah, I'll keep thinking about that. I like the concept. Other questions?
During the Ebola outbreak throughout several countries of South Africa, it was stated that there might be a resistance to Western nations sending vaccines in to Africa. Resistance because some belief that relating to other nations that there may be a problem, that we're actually causing the problem. I don't see this as, I see your solution as being potentially something that would overcome that. Because if you do it yourself, it's more readily acceptable by the indigenous populations. Have you seen that yourself there?
Yeah, so I'll tell you what I think that story is. So the question is, if we go into keep providing them with vaccines, I think that the concept is that the vaccines will mutate and we'll be getting other strains that our current vaccines aren't good against, and we're going to all be in trouble with that. So that's one of the problems with the fast rotation. The reason why I think this technology is so much better is because just like the cancer patient that we get the cancer, 4 weeks later we have the vaccine for that. Now you could scale up plants in these warehouses. Again, 1 day or 1 week you can do it. We can respond fast to any disease. So any changes, in theory, you could actually take that. If the hat's turned a slight brown, you can actually do it with that brown hat, if you will. But you can come up with a really rapid response, and that's where we have to go. We have to go to the rapid response. Right now, we just don't have the ability to do the response, as we all know, with the flu vaccine that's out there. We don't have the rapid response capabilities, and this is where we're going with this. So, good question.
Okay, you were giving 4 weeks as an example of how long it would take to develop. Is that a general amount of time that it takes to do 4 weeks, or is that like an ideal time, or the fastest possible, or?
So yeah, the question is, how long is this? When I say 4 weeks, could we really respond to H1N1 that quickly? And the truth is that the government challenged the company iBio, that's one of my colleagues, and they said, can you get a vaccine against H1N1? And here's the sequence that we know is there. How fast can you do it? 38 days. Okay? So, in theory, we're there. You know, in roughly a month time, you can actually get it. But they were challenged by the government. They actually give them lots of money, and they said, hey, for that kind of money, we'll make it. Okay? So the response is, the capabilities are there. But again, the part you have to do is, right now it's changing. It's a paradigm shift. You know, we've been doing it in eggs for I don't know how long. Okay? But now they're coming out with some other methods but they're very costly. This concept of using warehouses of these plants with these LED lights, I mean, that's like outer space, okay? You don't get people to change quickly to a new idea, but they're going to, okay? Because right now we're starting to worry more about this. And it's not when the next nasty flu is going to come, it's when, okay? We know it's going to come. I know many of you are familiar with the 1918, 1919 flu that killed in the range of 50 million people. That can come back again. That's still a possibility, especially with the global travel right now. We have to develop a rapid response, and the government knows it, and so the government's really getting behind this, and that's why I say if we can do an anthrax vaccine, because even if the fear on the anthrax vaccine, this is why I know the government is really into this, is if we get one of the bad nations coming in with a mutated anthrax that our current vaccine doesn't work on, even though it's frozen away somewhere, we're going to be out of luck again. So we need to be able to respond quickly because we can't wait multiple years to do it. We have to really talk about months. And if it's 2 months, okay, 3 months, even okay. But the faster we can respond, and the more plants we can have in these warehouses, and right now the number of plants that are out there for doing this kind of stuff is rapidly going up. I still like the ones that are in Texas with the glowy LED lights. I think those are just cool. Okay, I like pretty stuff. Okay.
So I wondered if you are getting any opposition from Big Pharma to this sort of approach, which kind of undercuts a lot of their business model, and are they trying to use patents and these kind of things to block this technology?
So the question is, what's Big Pharma doing about this? Yeah, Big Pharma doesn't like this, okay? So the technology for the cancer, Bayer bought that, okay? They bought it a lot of years ago, and we still haven't seen the vaccine out. So there's something going on there, but that's why the beautiful part about going to Africa is they don't care about Africa. They really don't. So we can develop it. We can get Africans to start making all their own vaccines, and once they can make it and make it cheaper, they can compete with our Big Pharma. So it's something I think we can deal with. And as long as we got people that are willing to go out and be the Don Quixote and try and go against the Big Pharma, and Big Pharma is good. I'm not saying they're taking care of a lot of diseases out there, but right now, and there is no incentive to cut the price, and so we need to give them an incentive to cut the price by giving some competition in that arena. Good question, though. One of our guests of honor is going to have to get it. You'll get it next.
Very unscientific guest of honor, but the patent concept. Science is supposed to be open and sharing. And then when you get a really unique idea, you can patent it. It would seem to me everybody's going to jump on this. Are they jumping on it? And are there other cancers that are being explored? This could just be so amazing and revolutionary. And I'm just wondering how universities and other scientists are reacting to this really new discovery.
So the question is the patent situation. How does that affect this? And so I got a really easy answer to that. Remember I told you I'm really old? I have 30 patents, okay? I got the rights for the developing countries for them, but a lot of them are starting to expire now. And so I'm giving it to every university I can give it to. I'm wanting the world to develop it. So I'm doing, a one-man battle out there because I know which patents are expiring and which ones have expired. And there's other companies that are doing patents on their new ways to make it fit better, faster, and stuff like that. But the old method is kind of coming off patent, so that's the good part about it.
The other part of my question was about other cancers.
Oh.
And is anybody tackling, there's so many out there, so many people so needy to have a specific antidote to their particular cancer. How is that being
Yeah, so
in the scientific community?
So this is a really good question because right now in the world of, and I've dealt with the FDA, so in the world of developing new therapies for cancer, do patents make a difference here? Yes, they do, but here's the real problem going here. The cancer therapy I showed you is a paradigm shift. Right now, the cost for the FDA average to approve a drug, even for the orphan drug diseases, runs you in the range of $600 million or so. Sometimes some say it's up to $1.3 billion to get a single drug approved. So the FDA has to change if you're going against small cancer groups and specific cancers like this one here. It turns out that again, the reason why we got through this with our clinical trials is because they have something that if you use the same process for all the things, so it's a procedure, they will approve it for the process. And so even though every patient had a different drug, we used the exact same process for it, and the FDA said they're fine with that. But so this paradigm, the FDA has to get kind of shift to be able to go to individualized therapy, and they are doing that. I mean, we're getting a little bit, with the Herceptin, they went with that because only about 25% of the women, 25 to 30% of the women have the HER2/neu antibody or a surface protein on their cancer. So basically, they have to check them out to see if they're going to be with this Herceptin drug is going to work on them. But I think that we can actually get down to really individualized cancers. In fact, right now, we're talking about basket cancers. So it turns out we used to always think lung cancer was lung cancer, breast cancer was breast cancer, cervical cancer was cervical cancer. It turns out cancers are related that aren't in the same tissue. So you may have a breast cancer that's identical in its surface proteins as one that's found in the lung, in a lung cancer. So what they want to do is identify using new analyses technique to find the surface proteins and go after use a drug that's against that cancer and not against the group as we all do it right now. The brain cancers, they have certain drugs they're using. Now we're looking, the FDA is okaying basket, and they call it kind of baskets because they're bringing all these different cancers that are related but on different tissues. And so we're going to see more of that going on. So the individualized, by knowing the genetic sequences of the cancer is going to come through. But again, I'm hoping that the technology stays open, but there's going to be definitely a lot of patent fighting going on in that area.
Thank you.
You had a question, didn't you? Yeah.
How did you get started with the tobacco plant?
So the question is, how did I get started using tobacco plant? You know, it's really simple. When I worked here in getting my Ph.D., the guy up the hall was the tobacco mosaic virus expert, Bill Dawson. He was absolutely awesome, and I loved talking with him, go out and have beer with him, and I was old enough, by the way. But got to learn a lot about them, and I kept thinking, you know, even though that wasn't my focus, I was working on viroids, but this concept, I said, this is just too neat because, listen, this virus goes into a plant. It takes over the plant and it's making this huge amount. I mean, basically, when it's in a cell, almost 50% of the proteins being made in that cell are the virus proteins. And you're saying, and the plant's okay. It doesn't die. And you're saying, that's awesome because if you had a virus making that much protein in your body, you'd be dead. Okay, but this plant's okay, we're okay with it. We look a little funny looking, you know, we may have funny looking fruit, funky looking lettuce leaves, but you know, plants are okay with viruses. Okay, very few die. There's a couple that they do die. Tristase is an example of it. But anyway, so the concept that there was this much made, this massive amount made in a plant, I go, what can we do with that? And so so I went and worked for a company, a pharmaceutical company for a few years, maybe 7 years. So it's a lot longer than I like to think. But and that's when I finally decided that we have to do something about this. And so with a colleague of mine, we went out to venture capitalists and raised the money to do it. But I was stuck on this ability of plants to produce this massive amount of protein, because proteins are good for disease fighting and vaccines and stuff like that. So that's how I got interested, right here in Riverside. You had one of the eminent tobacco mosaic virus virologists here.
Have you considered trying it for malaria, or would it even apply to protozoans?
Yes, so the question is, what about malaria? And yeah, so it turns out there is a protein that can be made. The problem with malaria, and the reason why I've stayed away from it, is it's kind of like HIV. It keeps changing, but it changes which proteins it shoves out on the surface. But apparently there's certain protein that they've had a hard time working with that it turns out plants can make. So this company iBio has gotten a grant to do them. Actually, I think it got the grant for Bill Gates to do it in plants, because they think the plants are the system for that. So that would be good, because we all know roughly 1 million people die per year of malaria. And I go to Africa a lot, and you have to take that Malarone or quinine water, or use a lot of DEET. One of those things that you need to do in order to avoid getting malaria, and I don't want malaria. So yeah, I think that will be coming up, and I think plants is going to be the right route to do it. I can't do all of them, so I have to let other people do it. I'd love to, but.
I'd like to ask a question that's sort of perpendicular to everything else. Now, I've known a lot of researchers who have gone to developing countries to save them or do a project and come back wringing their hands going, oh, it's impossible. You've been apparently very successful in Africa, and I was wondering if you could share a story or two about how you came to get to know and gain the trust of the people in Botswana.
Yeah, so the question is, how do I make this, working in the developing country is not the same as working here, okay? I was naive. I went there with this vision that I'm going to just start doing what I did here. You do this, you do that, let's do that, and come on, we're as a group, we're going there. It doesn't work that way, okay? You can either get frustrated and go walk away, but as Norm said, I kind of decided to stick with this. And I'll give you a really short example. When I went there the first time, I had told them, listen, I need this enzyme, I need this. I gave them a long list of the things I needed, because I was going to bring 4 students, and we were going to go through this whole technology, showing them everything in 4 weeks. We were going to make sure they knew it all. So I gave them the list, and then about 3 weeks before I come, I said, so do you have that stuff? Oh, no, not yet. I go, okay. Any problems? Oh, maybe. Okay, I need to know what maybe means. And they said, well, we're not sure we can get the money for it. I said, oh, I thought that we'd all agreed with it, But anyway, long story short, that wasn't really agreed on. So I said, okay, here's what I'll do. I'll bring everything but these enzymes that will fall apart if they're not kept at minus 40 degrees or minus 20 degrees, and they don't let you carry dry ice on the plane. So I said, you need to get that from South Africa and ship it up there. That's only $400. I'll buy all the rest. I'll put it in my backpack and I'll bring it. Okay? So I got 4 students. Now, it costs you $2,500 a student to fly them there. It's not cheap airfare. I have to take care of them while they're there, feed them, all this kind of stuff. So I have all these plans made. So we get there and I drop my backpack and I say, okay, let's get going. Oh, I go, what's the oh? Oh, we don't have the enzymes yet. I go, I thought we'd work this out. Well, anyway, long story short, I had to go through several layers of different people. You talk about bureaucracy. They have people who have people that talk to people for you, so like that. So anyway, I had to kind of work my way up and actually talk to all the different people one at a time. And it turns out you have to talk to them face to face. You can't do it on a phone. You can't do it by email. You have to go and talk with them, and then they really respond to you. That's what I learned the first time. But I still had this problem. How fast can you get it here? After they told me, well, we have to get this approved, and we have to get this approved by this person, and all this, and I even though I could walk it through, it was going to take 2.5 weeks. And I said, I don't have 2.5 weeks. So I'm what they call, I've learned how to speak Setswana, which is a language. So I'm what they call a Lahoa, which is an outsider. And so I can wrinkle things. I can kind of, kick some dirt, but I still have to work in their system. But as a Lahoa, I said, okay, I'm gonna fix this right now because I need it fixed right now. And so I found one of the graduate students, and I said, do you have a car? Yeah, well, he had to, it was a tough car, you could call it. He had a name for it. But anyway, his car had all kinds of problems, and I got wet in the car when it rained and stuff like that. But anyway, so I said, if I buy you dinner and lunch, will you drive me to South Africa, which is only 4 hours away? I'll buy it there with cash. You put your credit card in the ATM machine, it kicks out the money. And I said, I'll go buy it. And so the next day I went down to South Africa, I bought it, and we came back and we started the experiments. But I had to apologize for everyone for the next 3 weeks for kind of doing things not the way they do it. But I kept apologizing because I'm a Lahoa. Okay, so anyway, but now I know that I need to talk with them every year. I need to keep contact with them. I need the face-to-face meetings, so I will see the same people again. We go at a slower speed than what I would do here in the U.S., but we're having a great time doing it. And I think that now we're at the point we're getting ready for the clinical trials for the animal trials with the lumpy skin disease this year. So that's a breakthrough, and I'm really excited about it. And now I'm working also with people up in Kenya. They're excited about it. So as we go through one at a time, I think they thought I wouldn't stick with it because a lot of people leave, but I'm hard-headed. Okay, so I'm going to get it there, come hell or high water.
Okay, I think this is going to be our last question.
How do you get the vaccine made from the plants? What's that transition?
Okay, so how do we get? So basically, how do we get it from the plants? So that's a good question because it turns out, and I'm going to have to go talk about physical methods, but it turns out, again, I told you the great thing about tobacco mosaic virus is it's a massive amount of this protein. Even though it's decorated, it's still a massive amount. But it turns out all you need to do is chop it up like in a big blender, okay? And it is a big blender. And then you centrifuge it once to get rid, and that's a centrifuge that separates out the what I call cell debris. If you talk to someone who deals with plant cell walls, they don't call it debris. That's important to them, but it's not important to me. I like what's inside. So we spin out that debris, we save the fluid, and then now one of the biggest molecules in that fluid is this virus. So you run it at a really high centrifuge speed, and it smacks to the bottom of the tube. You pour the rest of it out, and then you have your purified. You're 97% pure there. You went through one last column. You got it cheap. So that's the beauty about it. You don't have to have this big, super expensive way of getting the protein out. Yeah. Good question, though. Are you making everyone stop?
Yeah. I want to thank the speaker. I want to thank the audience for coming. This was awesome.